Hey guys, this week's lecture is section A.2, or at least an abbreviated version of that. It's all about the human brain and how our nervous system interacts with the brain. I wanted to start with this slide that you see right here. It's kind of a fascinating story. This is a pair of med students in the 1920s who were particularly good at dissection. They were given the task by their instructor to dissect out every nerve, attempt to dissect out every nerve in the nervous system. And it's just a remarkable piece. Imagine trying to remove nearly microscopic cells out of a body without breaking them. It was pretty remarkable. Then they went in and they laid it on this piece of wood and put some little tags on it, stuff like that. It's on display in a museum right now, but I wanted to start here because I think it just kind of brilliantly shows the overall layout of our nervous system. We're looking at the central nervous system. Here's part of the brain moving down into the spinal cord. All that is our central nervous system. All of these nerves branching out from the center is our peripheral nervous system. And so if we're going to get started here talking about our nervous system, we need to get that straight right off the bat. Central nervous system, brain plus spinal cord. Peripheral nervous system, everything else that branches away from the CNS. I'll be referring to them as the CNS or the PNS. That's central versus peripheral nerve system. And so if we look at our central nervous system, it's just the brain and the spinal cord. You can consider this sort of like the command center. The peripheral nervous system is involved in carrying signals to and from the command center. So we're going to be carrying messages to the brain and spinal cord. These are going to be sensory messages, like, ouch, I'm hot. And then going away from the central nervous system, back to the arm, move your hand, it's hot, etc., etc. So let's take a second and take a look at the peripheral nervous system. It consists of basically two different pathways. We have our conscious movements and our sort of involuntary responses. Those involuntary responses are called our autonomic responses to a stimulus. Think of jerking your hand away from a hot stove. Whoa! Those are involuntary. But more than that, you also need to consider things like your heartbeat being regulated. That's an autonomic response. The contractions in your GI tract, so your small intestine contracting to move food along, all that is involuntary, autonomic responses. Somatic responses, on the other hand, those are our voluntary responses, like food, nom nom nom, reaching out for your sandwich. It would be a somatic response. And so both of these are different varieties of neural pathways in your peripheral nervous system, in the PNS. Let's take an example. This strikingly, I don't know where he's looking because his glass of water is down here, but let's say he's looking at a glass of water. His eyes are going to send some sensory input through to the brain. And so these nerves are going to carry the sensory input to the brain. And then it's the brain's job to integrate the sensory information. Now, integration is an important word here because you have to understand that, yes, his eyes are sensing that glass of water, but the next moment is that he's going to need to reach for that glass of water. And so there's also some sensory input about his sense of space and how he's going to coordinate his movements. So all this information about space, visual input, is then going to be integrated in different regions of the brain. And then the brain will send a message out to a motor neuron. So we have sensory neurons going in, motor neurons going out. So our motor output will then synapse with muscles, which move the arm to bring the glass of water to his devilishly handsome lips. And then he can have his drink of water. So this is a typical example of a somatic neural pathway. If we jump back to autonomic pathways, these are our involuntary processes, and most of these will connect with not the cerebellum, or the cerebrum, but mostly they're dealing with our brainstem, this region right here. And within the brainstem, there are specialized regions for different functions in the body. We discussed one relating to heartbeat, which is in the medulla. So this is the brainstem, just dissected out. You don't need to know the individual parts of the brainstem because, well, your exams are canceled. But here are the different regions. I will reference these moving forward. The one that we've already mentioned was the medulla. Now the medulla, if you'll remember from the heart subtopic, has the cardiovascular center located in it. And so there are nerves that connect to the medulla, which are specifically related to controlling the heartbeat by regulating the pace of the SA node, the sinoatrial node. And so breathing and heart rate are regulated by the medulla. So here's going back to our heart heartbeat or heart cardiac output. So remember, out in the body, there are all kinds of sensors. Pressure sensors to test for blood pressure. There are pH sensors to test for CO2 concentration. And so with these sensors are going to then send sensory pathways to the medulla, part of the brainstem. And so now the medulla has to integrate that information and determine whether the heartbeat should be increased or decreased. Then it sends messages through motor pathways down through the spinal cord. Here's our spinal cord. And then out to the SA node to modify that heartbeat. So this is an autonomic pathway, part of our peripheral nervous system. So these nerves are coming from, in this case, this is a pressure sensor in the aorta. There are pressure sensors elsewhere, which can send sensory information to the medulla. And then that motor information can be sent back to the heart to either increase or decrease the heartbeat. And so what we've been talking about is how sensory information can get to the brain and then go back out to form an action. So sensory neurons connecting to the CNS, the central nervous system, and then motor neurons giving you your action or your response. Occasionally, in very rare occasions, and we'll talk about this a little bit later, you can get what's called a spinal reflex arc, where the brain is skipped entirely, and you need a very quick response. And so a sensory neuron will then just synapse through some neurons in the spinal cord, skipping the brain entirely, and then go directly to a motor neuron. So this is that sort of jerking reaction that you get when you have a reflex, like the patellar reflex, where they tap on your knee and your knee just kicks out. That has to do with a spinal reflex arc. Here's another example. Don't touch fire, fire bad. But if you were to touch the candle, you'd receive some sensory information that's hot, ouch. And so that sensory information then goes into your spinal cord. It's going to bypass the brain. It's going to make a very quick transmission going through the spinal cord. Here's a close-up on the spinal tissue. And then relay that information out through a motor neuron out to the muscles, so you can very quickly, instantly, and without thinking, move your hand away from that pain, from that heat source. Now, how does your finger know that this is hot? Well, there are tiny little receptors for things like heat, pressure, pain in your fingers. Those synapse with neurons send that sensory information to the CNS, the central nervous system, in this case the spinal cord, not the brain. And then the spinal cord will then synapse with more motor neurons, which get your hand out of the way, hopefully. Another reflex, again, this is a reflex, but be careful, this reflex is not a spinal reflex arc. This one does innervate, if you will, with the brain. We're looking at the pupillary reflex right here. And this is, I'm sorry, this is disturbing, this continuous dilation and contraction of the pupil. Sorry, just nail that. Okay, so here we go. Quick anatomy of the eye. What we're looking at here is obviously the human eye. We've got the iris, which is where the coloration of the pigment of the eye is located. Now, the iris also has some circular muscle in it. That circular muscle can dilate and contract. And when it contracts, it squeezes to a tighter ring like this. And then this blank space in the middle, this is the pupil, is now smaller. And so what what this does is it prevents an excessive amount of light from getting into the eye that can cause serious damage to the back of the eye called the retina. So this pupil is just a hole into the empty ball, which is your eye. And so when light is present, the pupil contracts. When it's dark, the pupil will dilate. And that dilation allows more light in. Now, if you have a bright light and it's shining in one of your eyes, both of your irises are going to contract together. And here's why. Because the pupil reflex pathway is rather interesting. Your eyes are connected to two nerves called the optic nerves. Now, these optic nerves will carry themselves back through the skull, and then on the inferior portion of the brain, underneath the brain, there's a spot where they cross each other, and they form an X. This is called the optic chiasm. And so at this X, these optic nerves will actually cross each other. And what this does is it allows one eye to send information to both sides of the brain. This is, right here, this is the midbrain, which is involved in the pupillary reflex. And so what happens is, if you shine the light in one of your eyes, that information gets split at the optic chiasm, gets integrated in the midbrain, and then motor neurons are going to send information out to both eyes, so that both eyes will contract. The iris, if you will, of both eyes will contract at the same time. So if you did the same thing in the other eye, if this light source were to be moved to the other eye, both eyes, again, should still be contracting. If you shut the lights off, both eyes will dilate once again. And so this is called the pupillary reflex. And this, as you know, if you've done your homework, it can be used to test for brain damage, because if there's damage to the brainstem, the midbrain, you are in deep trouble, because the midbrain and the rest of the brainstem can be used to control all of your autonomic functions, which means things like heart rate, digestion can be completely screwed up. So if you want to check for serious damage to the brain after a traumatic injury or perhaps a concussion, this is a very simple way to look for severe damage to areas like the midbrain. And so this is the pupillary reflex. So after a severe head injury, one of the first tests that you can do with something as cheap as a light bulb is to check for a normal pupillary reflex. Okay, so that's the basic structure of your central nervous system, your peripheral nervous system, how they communicate with each other. And then moving forward, for the rest of the lecture, we're going to focus on the structure of the brain and specific functions. I'm going to try and make this quick and relatively easy because you don't have exams. And if you want more detail, feel free to read the section A.2 to fill in those gaps. So, yeah, look at that. So starting with the centerpiece of the human brain, this is the structure that makes us so special. It's our cerebral cortex. It's this sort of transparent stuff in this GIF on the side over here. And the cerebral cortex in humans is the largest portion of the brain. It has a lot of infolding. You can see all these little folds in here. These are actually just part of one continuous sheet. So imagine if you had a big sheet of tissue, which then gets folded up into all of these, grooves and ridges, and that's what we're looking at here on the outside of the cerebral cortex. Why would we want to fold it? Surface area, Okay, and this is where we do all of our higher order functioning, like reasoning and decision making, stuff like that. So if we were to look at the overall size of the brain, most of it's about the same for most species. The big point where we diverge is in the size of our cerebrum or our cerebral cortex. But if you were to take the overall size of the brain compared to your body mass, there should be a relatively linear correlation here where we have bigger body, bigger brain. And then intelligent creatures will, of course, be on the positive side of this line here. We've got Homo sapiens right there with a relatively large brain for our bodies. Dolphins are here as well, chimpanzees. Elephants really aren't... Anyway, If you go below our line, this is where their brain is abnormally small, considering the size of their body. Things like alligators, the Tyrannosaurus, we think, whales. So there is generally a trend where bigger body, bigger brain, unless you're a particularly smart animal, or you rely on more reasoning, if you will. You'd have a bigger cerebrum. That puts you above this line. So the cerebrum is divided up into 4 lobes. You can see them here: the frontal, parietal, temporal, and occipital lobes. Now these lobes each have special functions because there are specific regions in these lobes which are designed to carry out special functions. Now, when you do any one task, like for example me speaking right now, in order for me to speak, there's the mechanical movement of my mouth. There's the thought process, the logical reasoning of the words that need to come out of my mouth. There's the auditory understanding of the sounds. And of course, in some cases, I might be reading as I'm speaking, so there's also a visual component associated with speech to some extent. And so any simple task, like speaking, could theoretically involve all 4 lobes of the brain, because there's different aspects to things like speech. So we'll talk about functions, but you have to keep in mind, for any function, you're integrating multiple different regions of your cerebrum. So here we go, starting with the frontal lobe. The frontal lobe obviously is in the front or anterior portion of the brain. It contains the motor cortex. So our voluntary movements must be decided upon by our motor cortex in the frontal lobe. Now, some of our balance and posture and that kind of stuff is also helped by our cerebellum back here, but the decision to make a particular movement or a different movement is all done with our frontal lobe. Also located in the frontal lobe, as you know from the homework from last week, 2 weeks ago, is the nucleus accumbens, and this is right here, the little red dot. It's in the frontal lobe, down here in the bottom of the frontal lobe. This is involved in dopamine pathways, and if you don't know about dopamine, it's a neurotransmitter that gives you a sense of well-being or happiness. And so what the nucleus accumbens will do is release dopamine when you deserve a reward. So if you do something good, then this region will release dopamine. So it gives you a sense of positive reinforcement, like if you're gambling and you're anticipating a reward for your hand, that you want to win some money or whatever, this dopamine center will help start to release the neurotransmitter dopamine. That the nucleus accumbens will start to encourage the release of dopamine. So you'll feel good about your choices. Unfortunately, drugs will do the same thing, and this can lead to addiction, which is why people can become addicted to anything involving a reward, like gambling, or even things like exercise or other things. And of course, drugs are the worst case scenario because they artificially release these, happy dopamine neurotransmitters, and we'll do a lot more on how drugs work later on. But for now, this region, the nucleus accumbens, gives you a reward for positive behavior. Sometimes that positive behavior is not so positive, but that's okay. And then in the frontal lobe, we also have the Broca's area, which is involved in some mechanical aspects of speech. I'd like to take a quick side detour and just show you a quick video of what happens when your Broca's area is damaged. This is a gentleman who for whatever reason, is not getting proper communication to and from his Broca's area. So the mechanical aspect of his speech is damaged. He has the ability to think about the words that he would like to say, but the mechanical production of those speech, those words, it just does not connect. Okay, let's take a look.
Name?
Mike Caputo.
And Mike, when was your stroke?
I was 7 years ago.
Okay.
And
what did you used to do?
Worked Autodesk, seven sales, and worldwide, and very good. Yeah.
Okay. And who are you looking at over there when you turn your head?
That's my wife.
Okay. And why is she helping you to talk?
She's a speech
So you have trouble with your speech.
Yeah.
And what's that called?
Aphasia.
All right. And so why don't you work now?
I do.
And what do you do now?
Voices of Hope aphasia.
And what is Voices of Hope?
Peterburg, and Dr. Hinckley and myself, founder for me, and I members the members probably 60 people.
So 60 people are part of Voices of Hope,
Yes.
which is an aphasia support group
Yes.
that you founded.
Yes.
And Dr. Jackie Hinckley is part of that.
Yes.
Okay.
Yeah, it's very difficult to watch, but that's an example of what's called Broca's aphasia, where the ability to speak is lost because of damage due to a stroke to the Broca's area. Moving on, we have the next lobe called the parietal lobe, which contains the somatosensory cortex. This is involved in your spatial sense, including your visual sense of space. And so when you're reaching out to touch something or to click a mouse or type, it helps to coordinate your awareness of your physical space along with the visual aspect of the world around you. It also helps to coordinate some movements that are involved in speech, again, because speech is both a physical and an auditory activity. Moving on to the temporal lobe, this has the primary auditory cortex. It hangs right by the ear, so it might be easy to remember that. So sound is processed here. And this, along with Broca's area, can help with language recognition. In addition, a little bonus function of the temporal lobe is that there's short-term memories stored there until they can be either stored as long-term memories or sort of washed away. Then move on to the occipital lobe in the back, the posterior portion of the brain, which maybe surprisingly is where the visual cortex is located. So those optic nerves from the eyes dip under the brain and then connect back in the back where the occipital lobe, right back here. Visual input is processed there. Now, again, as I said up front, most of the functions that you do at any given moment require a sense of space, probably rely on some sort of sight or sound to integrate all of this. And so usually your four lobes are integrated into any given function. Now, in addition to integrating between the lobes, you have a right and left hemisphere of your brain, and those must be integrated. And so you are sending signals constantly back and forth between the right and left hemisphere through this bundle of nerves right in the center called your corpus callosum. And so perhaps you've heard this before, but and my camera is on mirror, so my right and left might be switched. So I'll just go with, instead of right and left, I'll say one side and the other. So if I'm getting a sensory information from this side of my body, chances are the other side of my brain will process that. So your brain processes information from the opposite side as far as the sensory input. And that communication across those hemispheres takes place at this region, this structure here, bundle of nerves called the corpus callosum. If you just consider the eyes for a second, right here, this is your left eye. Over here in green, this is what's called the left visual field. This is, if you were to take the midpoint of your face, this is the left side, so the left visual field, the things you see on the left, technically the things you see on the right over here. Now you have to keep in mind that your left eye can see things that are straight ahead. It can see things that are just off to the right. It can see some things that are way off to the right. And so your left eye will see some things in your right visual field, mostly things in the left visual field. And your right eye will see mostly things in the right visual field, and then some things in the left visual field. And what happens is, again, remember these optic nerves and the optic chiasm where they cross over, they're going to be sending information from the right visual field over to the left side of the brain. Keep in mind that the right visual field can be seen by both eyes, and so this part of your eye sees things over on the right visual field and then sends things over here onto the left side of your brain. And then this eye, your right eye, can sense things over in the left visual field, sends those over to the right side of your brain, and then mostly sending information here from the right side crossing over to the left. Over here, this is mostly sending information crossing over to the right. So your eyes basically send information to the opposite side of the brain to interpret. The next video I'd like to check out with you guys is this person who had severe seizures due to a condition called epilepsy. They were debilitating. He could not really get through a normal day. So he went for a very intense operation where they go into the middle of the brain and they sever the corpus callosum. And so the left and right communication has been broken. But for whatever reason, this helps with the seizures involved with epilepsy. So this video shows exactly how that right-left hemisphere communication breakdown can affect things like drawing or writing or interpreting words and shapes. So here we go.
Show you some things. I just want you to tell me what you see. And here we go. You ready? Look right at the dot.
Okay.
Right. Okay, you ready? Look right at the dot.
Grapes.
Good. When Joe focuses on a point, look right at the dot, everything to the right of the point goes to his left brain, the dominant hemisphere for language and speech. Look right at the dot. So we can see here that when we flash a word or a picture,
Tree.
Joe is easily able to name it. Here we go.
See it.
Close your eyes and let your left hand do a little work here. Okay, what do you got there?
Pan.
Okay, very good. Now, when a word or a picture falls to the left of a fixation point, that information goes to his disconnected right half brain. And as we can see here, Joe is unable to name it. Joe is able to draw the picture with his left hand, the left hand getting its major control from the right half brain. What'd you draw?
Okay.
What'd you see?
Wheel on one side. I don't know where I saw on the other.
So even though he can't name it, his left hand is able to draw out a picture of the stimulus of the picture or word that we presented to his right half brain. What did you see?
Saw a hammer.
So just close your eyes and draw with your left hand. Just let it go. That's nice. What's that?
Saw.
Yeah. What'd you see?
Hammer.
What'd you draw that for?
I don't know.
What we have.
Pretty fascinating. So as you saw with Joe, the communication between his left and right hemispheres was broken, but the individual functions of both the left and right side of his brain were still intact, and so he could do individual activities, but communicating with each other was no difficult. Kind of an interesting, it's called split brain condition sometimes. It's sort of the informal name for it. Moving on, so the last thing we're going to do, and this is quick, is just, get this out of here, just quick breakdown of some of the ways that we study the structure and the function of the brain. The easiest way, of course, is animal experimentation, but that's not very ethical and it's not nice when you think about it. Of course, we have autopsies. Autopsies are important if somebody suffered some kind of behavioral condition that was very unusual. They had some sort of damage to the brain that was maybe unnoticed. Then after they die, we can look at the brain, the structure of the brain through dissection to see exactly what was different from a typical brain. We can also look at brain lesions through autopsies. And then the most common way to actually study the brain structure is through what's called an MRI. Or if we want to look at how the brain works, we can look at an fMRI. And I want to show you the differences between those two. Right here on this slide, we have an MRI. Now, an MRI is basically just a still photo, and you can only get one layer when you're taking a photo. So notice this is deep inside the middle of the brain over here. And you can see some structures, corpus callosum, there's the thalamus in here, the frontal lobe, occipital lobe back here, cerebellum, brainstem, and spinal cord. And so this MRI allows you to take a still shot of just the structure of the brain. So if you're looking for something like a tumor or some sort of physical abnormality in the brain, this is a perfectly fine way to do that. And you can take them from all kinds of angles. So from above, here's another shot, an MRI. You can see these eyes, the ocular nerves, optic nerves, dipping back, and then they would under back to the occipital lobe. So another picture of an MRI taken from above. You can take a photo and then MRI. This is called a sagittal section. You can look at the middle of the brain from this perspective. Again, we're looking for abnormalities in the structure of the brain. I'm not going to tell you a whole lot about function yet. When we go to an fMRI, the F stands for functional or function. So here's an fMRI. And the way this works is an fMRI is going to track the movement of water, more specifically the movement of blood, through different regions of the brain, because hopefully you're aware by now that the brain, since it's made up of about 86 billion neurons, requires a ton of energy because every neuron needs ATP for active transport. So we need a lot of energy, which means we have a lot of ATP. So blood flow is very important. And so we shuttle blood to different parts of the brain to keep those neurons moving, to keep a constant supply of glucose for ATP production, et cetera, et cetera. Anyway, so as you use different parts of your brain, different blood vessels will open and close to allow for the movement of blood to and from different areas. And so we can look at brain activity by tracking the flow of liquids throughout the brain, and this is how an fMRI works. So in this picture, we're looking at the regions of the brain that are lighting up on the fMRI when a person is speaking. You can see there's a few different areas involved with speech. Temporal lobe, we've got some speech and Broca's area working over here. This is a person just tapping their finger, and here's a person listening. Notice there are multiple regions lighting up. This one's interesting. This is an fMRI of a person who was gambling, basically, and then they, in the research study, they basically artificially gave them good cards right before they took this fMRI, and so they're anticipating a reward. So what region of the brain is lighting up? I'm not going to tell you. Look at your notes. And so this is kind of an interesting one. So an fMRI can be used to look at the functioning of the brain. All right, guys, that is what I have for you. So moving forward, you should now know the basic structure of the central and peripheral nervous system, some basic structures of the brain and how they communicate with each other. And then moving forward into the next few sections, we're going to talk about how our bodies actually perceive information, how the eyes and ears can pick up information and then send that to the brain. And then we'll talk about neuropharmacology, so how different drugs can affect brain behavior. And then we'll wrap up with some actual studies of behavior and how behaviors can be changed. And so that's what I've got. Hope you guys had a great week, and I'll talk to you soon.